Okay, so we'll start with part A of chapter 11, which is the nervous system. And it goes through some of the anatomy that hopefully, I'm hoping you guys, this will be a review from anatomy a little bit. We've skipped chapter 10 because 10 was just on nomenclature of muscles. So the next couple chapters are going to be all about nervous system, which everyone loves, but we're going to try to get a good base here using chapter 11 as our starting point. Your nervous system is your master control and communication system of the body. And all of your neurons, the cells in the nervous system, will communicate via electrical and chemical signals. These will be very rapid and specific signals, and they usually cause immediate responses. The nervous system has three overlapping functions. There's the sensory input, the integration, and the motor output. And the sensory input will involve gathering information from sensory receptors, traveling through sensory neurons up to an integration center, which is usually in your central nervous system, which is your brain and spinal cord. Your brain and spinal cord process and interpret all of that sensory input and then send out some sort of motor response. And that motor response will usually activate some sort of effector, whether that's a muscle, a gland, or another organ. So this is the basic of the nervous system. Let's see, you're looking at a glass of water, or you're looking at your computer screen right now, or you're listening to my voice. That sensory input gets sent up to the integration center. It can be your brain, but sometimes sensory input goes just to your spinal cord and goes back out to the motor effector. So often it does go up to the brain integration center, but it can just go to the spinal cord in what we call reflex arcs. And then the integration center, whether that is the brain or just the spinal cord, will send out some sort of motor response. We divide your nervous system into your central and peripheral nervous system, the brain and spinal cord and your CNS, and then all the peripheral nerves in your PNS, your spinal nerves, your cranial nerves. So here's a look at the divisions of your PNS versus your CNS. The sensory afferent division, so this will be the division that collects all sensory impulses from the exterior and interior environment of the body, we divide it into somatic sensory fibers, which will take impulses from your skin, your skeletal muscles, and all of your joints to your brain and spinal cord, or visceral sensory fibers. Whenever you see the word visceral, that has to do with organs. So these will convey impulses from your visceral organs to your brain and spinal cord of the CNS. Then your motor, or your efferent division, transmits impulses from your CNS to all the effector organs, whether they are muscles or glands. And we divide the efferent division into your somatic nervous system, which goes to all skeletal muscle, and your autonomic nervous system. So you should have a good understanding of these two divisions because it's kind of the basis for the rest of what we'll talk about with the nervous system. So your somatic nervous system takes any sort of motor responses to all of your skeletal muscle. So this involves voluntary control and conscious control of your skeletal muscles. Your autonomic nervous system consists of visceral motor nerve fibers. So they'll go to your viscera or your organs, anything inside body cavities, your smooth muscle, your cardiac muscle, your glands. The ANS is involuntary. So it's autonomic or automatic. Think of that. It's involuntary. These are the two functional divisions of the subdivision, the sympathetic and the parasympathetic division. And if you had me for anatomy, I like knowing the differences between these two, and I'm going to keep it that way in physiology too, understanding the differences between your sympathetic and your parasympathetic nervous system. They will work in opposition to each other, sometimes at the same time if they're trying to increase or decrease something. So here's your central nervous system. So in blue, you have any sort of sensory input to your central nervous system, and then your central nervous system integrates that and sends out a motor response through your cranial, spinal nerves, and then it will go into your peripheral nervous system into the motor or efferent division. So these will be motor nerve fibers. They will be going to your skeletal muscles in the somatic nervous system, conscious voluntary control, or the autonomic nervous system, involuntary visceral control. and we separate the ANS into the sympathetic and the parasympathetic division. Sympathetic division, does anyone remember the kind of the name or nickname we gave that?
Sympathetic was rest and digest.
Close.
Fight or flight?
Yes. Good job. You got it on the second try. Good. So yeah, sympathetic is fight and flight. So this prepares your body for all emergency type situations. So stressful situations, it'll dilate your pupils, increase your heart rate, decrease activity to your gut because you're not worried about going to the bathroom when you're in a really stressful situation, or you're not worried about digesting your food because some people do, lose all control during stressful situations as well. And then the parasympathetic division, the rest and digest. So I say this is the division that's active after Thanksgiving. Okay, the histology of the nervous system consists of neurons, which are your nerve cells, and then the neuroglia, which are all the surrounding supporting cells. These are the neuroglia support cells of your central nervous system. So these 4 neuroglia are only in your brain and spinal cord. Astrocytes are the most abundant. Astrocytes function to support and brace neurons. They will control the chemical environment around neurons. They'll participate in information processing in the brain. This is a fun look at an astrocyte. It has what we call perivascular feet, which wrap around capillaries. So it helps to form what we call the blood-brain barrier. So it just controls what comes out of capillaries and goes into your brain interstitial fluid. So that's an astrocyte. Microglial cells are part of kind of the immune response. They'll transform to phagocytize microorganisms. So they'll get rid of anything harmful. So these are examples of microglial cells. They're a part of your defense cells in your brain and spinal cord. Ependymal cells, these may be ciliated. They form a barrier between cerebral spinal fluid in cavities and tissue bathing fluid cells and tissue fluid bathing cells in the central nervous system. They will have cilia that will help to beat and circulate cerebral spinal fluid. So here are the ependymal cells. They're filled with cerebrospinal fluid, or they form the barrier between the fluid-filled cavity and the brain itself. So these are the ependymal cells. Oligodendrocytes are in the central nervous system, and they form myelin sheaths in your central nervous system. Oligodendrocytes form the myelin, which helps to electrically insulate the axons of neurons. So it helps the electrical stimuli to travel faster through the axons. So that's what myelin does. The 2 main neuroglial cells of your peripheral nervous system, and again, a neuroglial cell is just a support cell to a neuron, are satellite cells and Schwann cells. The big one here is a Schwann cell, also known as a neurolemocyte. They will surround all of the nerve fibers and form myelin sheaths in the thicker nerve fibers. So Schwann cells are to oligodendrocytes. So Schwann cells are in your PNS, and oligodendrocytes are just in your CNS. So here's a look at a Schwann cell, and they will form the myelin sheaths around a nerve fiber. Here's the satellite cell, helping support the cell body of a neuron. All right, neurons themselves, they will be specialized cells to conduct electrical impulses. Some cool things about nerve cells: neurons, I'll use those words interchangeably. They have an extreme longevity. They will last your whole lifetime, which is kind of cool to think about. They are amitotic, meaning they don't divide, and they have a high metabolic rate. So they need a continuous supply of oxygen and glucose. All of them will have a cell body. The cell body is also called the soma. The soma is where it'll synthesize all the proteins and chemicals it needs. I just want you to know that a neuron has a cell body. Most neuron cell bodies are located in your central nervous system as nuclei, which are clusters of neuron cell bodies in the central nervous system. And if you see the word ganglia in the peripheral nervous system, a ganglia is just a cluster of neuron cell bodies. So we call it a nuclei in the central nervous system and a ganglia in the peripheral nervous system. And each just mean a cluster of these neuron cell bodies. It has arm-like processes, which are dendrites and axons. A tract is a bundle of neuron processes in the central nervous system. So you'll hear of different tracts and their names, but that's just a bundle of these processes put together like a bundle of axons. And we call a nerve a bundle of neuron processes in the peripheral nervous system. So when we name a nerve, or you remember nerve names from anatomy, you're naming basically just a bundle of axons or dendrites that have been bundled together. So this is a typical structure of a motor neuron. Here are the dendrites, which will receive, they'll have the receptive regions. The cell body will process, integrate everything that the dendrites receive, and then it'll send it on down the axon. The axon is covered with these myelin sheaths. They look like little blue marshmallows. They're the Schwann cells that are creating the myelin sheath. So the Schwann cell is within that. The myelin sheath gap is important because the electrical impulse will jump between myelin sheaths as it travels down. And the axon hillock is the location of where the axon joins with the cell body. And this is the initial segment of the axon. This is where an action potential will begin. And an action potential is just the name for a firing of a nerve. So if we have an action potential that travels down this axon, it means it's carrying an electrical signal to some part of the body. So that's what an action potential is. It's just an electrical signal through an axon. This takes you through what dendrites are, the receptive regions. Here are dendritic spines leading all to the neuron cell body. Here is the axon, the structure. The axon hillock is the beginning initial part. The distal end of axons are called axon terminals. Functional characteristics of the axon, this will just be the conducting region of the neuron. At its terminal region, it will secrete what we call neurotransmitters, which will be released into a space before that neurotransmitter will bind to a receptor on either a muscle cell or another neuron. They can excite or inhibit neurons that it contacts, and axons can carry on many conversations with different neurons at the same time. So they're really cool structures of nerves. They can quickly decay if cut or damaged, but Schwann cells will help in the regeneration of axons itself. Some functional characteristics of axons, they have efficient internal transport mechanisms. Movement will occur in both directions on an axon. Anterograde is away from the cell body. An example of this will be in mitochondria, cytoskeleton elements. Just you need to know what anterograde and retrograde is. Retrograde is back toward the cell body, and these are organelles to be degraded signal molecules, viruses, or some sort of bacterial toxins. So normally most processes out of the cell body will go anterograde, away from the cell body. If it's retrograde, it's a movement of that electrical signal traveling back toward the cell body in the axon. And this is important for some clinical aspects. Certain viruses and bacteria damage neural tissues by using retrograde axonal transport. So certain toxins and viruses travel through the axon up to the cell body to damage that neuron in the cell body. An example is polio, rabies, herpes, other viruses, a tetanus toxin. And there's some research underway using retrograde transport to treat genetic diseases. Okay. The myelin sheath. The function of myelin in general is to protect and electrically insulate the axon, and it just helps to increase speed of the nerve impulse transmission. Myelinated fibers will have segmented sheaths surrounding most all of them, or their large diameter axons. These messages travel faster in larger diameter axons, and they will be myelinated. Non-myelinated fibers do not contain that myelin sheath, and they will be a little more slow in conducting that electrical impulse. In the peripheral nervous system, your Schwann cells create the myelin sheath. That's about all I want you to know about that. This is what myelin looks like as it's forming around the axon itself. Here's the myelin sheath around the axon. Myelination in the peripheral nervous system. There are gaps between sheaths, and we call them formally node Ranvier. He must have been the guy to identify them. Non-myelinated fibers might still have Schwann cells, but they won't coil up in the myelin. Okay, so that's another structure just showing the Schwann cell wrapping the axon in myelin and the little gap, the nodes of Ranvier. The central nervous system forms myelin by the oligodendrocytes. And that's all I really want you to know about that. The difference between white matter and gray matter in the central nervous system, you should know this, what makes matter in your CNS white and what makes it gray. The white matter will be regions of your brain and spinal cord that have those myelinated fibers. Myelin is white, so it will color the brain and spinal cord tissue white. They are usually fiber tracks, so that means it'll be some sort of pathway to and from the brain. Gray matter are mostly neuron cell bodies and non-myelinated fibers. So in your brain and spinal cord, you have parts of your brain and spinal cord that look gray, and they are made up of cell bodies and non-myelinated fibers. And again, this is just because myelin is white. Okay, here are oligodendrocytes creating the myelin sheaths in your CNS. Classification of neurons, they can be multipolar, bipolar, or unipolar, and they describe, these are the structural classifications just based on the number of processes they have. A multipolar would have an axon, other dendrites. Bipolar would have one axon, one dendrite. And unipolar would be like one T-like process with just two axons. And this is a look at what the structural classification of neurons would look like. Again, the differences between a multipolar, bipolar, or unipolar. Not too excited about knowing that, but probably is good to know. Comparisons of more structural classifications of neurons kind of goes through again, giving you an example of a multipolar, bipolar, and unipolar, what they might look like, how they would still receive a signal from the receptive ends and still have it travel off through a cell body that's kind of off to the side and down an axon. So different structures of neurons, but the one that we're really going to be focusing on a lot is the multipolar, the one that you're probably familiar with from anatomy. Okay, functional classifications of neurons, they can be sensory or motor. Sensory just means they take impulses from any sensory receptor, they take it to the brain and spinal cord. Almost all of these are unipolar, and the cell bodies are located in ganglia. The motor neurons, most all of them are multipolar, and they carry impulses from your central nervous system to all the effectors. When we talk about an interneuron, this is the third type of neuron that I want you to know. These are just the link between the motor and the sensory neurons. Actually all of the time, interneurons are located within your central nervous system. And there'll be just a place of synapse or a junction to combine a sensory to a motor neuron. There'll be an interneuron between the two. It's interesting to note that 99% of your body's neurons are interneurons. So kind of these, I call them the mediators. So they're ones in between. Okay. These are functional classes of the neurons and examples of each. If you're interested, we talked about unipolar neurons are mostly sensory neurons, and most of our multipolar neurons are all the motor neurons. An example of and this gives you an example of a bipolar neuron. A good example of this will be when we study your sensory system in the eyes, we'll see bipolar neurons there. So that you'll come back to learn more about that. Okay, questions on this part A. Okay, I'm going to start part B just to get started on it. Let's see. I don't want to give you too much information tonight, but we'll start part B if I can find it. Okay. All right, so hopefully you're seeing part B in front of you. And part B talks about action potentials, which is how electrical signals get passed through your neurons. So membrane potentials. Like all cells, and we talked a little bit about this with your muscle cells, all neurons also have a resting membrane potential. And this refers to the electrical charge within that cell right underneath the membrane. Unlike most other cells, neurons can rapidly change their resting membrane potential. Neurons are highly excitable, which is a good thing. You want your nerves to be able to respond to stimuli and send down an electrical signal instantaneously. The basic principles of electricity are that opposite charges are attracted to each other, and energy is required to keep opposite charges separated across the membrane. Energy will be liberated when the charges move toward one another, and when these opposite charges are separated, the system has what we call potential energy. So when we talk about voltage, because we'll talk about the membrane voltage, this is just the measure of potential energy generated by separated charge. And it's measured between 2 points, and we'll often label it as millivolts. It's called the potential difference or the potential. So this is why we'll call it the potential, the membrane potential, because it's potential that exists between this energy separated by the charges. The charge difference across the plasma membrane results in potential, and the greater charge difference between the points is equal to a higher voltage. Some more definitions, and this will all make more sense when we put it all together. The current refers to a flow of electrical charge, and in your neurons, we'll be dealing with an ion, which is a positively or negatively charged atom between 2 points. And this current can be used to do work as it flows. Resistance is any sort of hindrance to charge flow. Ohm's law, we're not going to get too much into this, but it gives the relationship of voltage, current, and resistance together. I'm going to pass over that because I just want to get to the role of membrane ion channels. Within the membrane in neurons, you have different ion channels that allow ions to pass through them. These ion channels are large proteins. They serve as a selective membrane ion channel, so only certain ions can pass through them. The potassium ion channel only allows potassium to go through. There's two main types of ion channels. They can be leakage channels that are always open, or gated channels in which part of the protein changes shape to open or close that channel. It can be chemically gated, so a chemical has to bind to that channel to open or close it. Voltage gated, meaning an ion with a voltage has to pass through it, or mechanically gated, something has to mechanically open or close. The chemically gated are called ligand gated channels, and they open only with the binding of a specific chemical, like a neurotransmitter. So we talked about acetylcholine binds to chemically gated channels in your muscle cells. Voltage gated channels open and close in response to changes in membrane potential. So when an electrical charge or an action potential travels through a neuron, it will open up voltage gated sodium or potassium channels. And a mechanically gated channel opens and closes to some sort of physical deformation of receptors. So this is an example of a chemically gated ion channel. Like with neurotransmitters, voltage gated ion channels will be open or closed in response to changes in the membrane potential. And that's what we're going to focus on tonight with action potentials. These are talking about gated channels and how ions will diffuse quickly when gated channels are open, when their concentration gradients are higher, or toward an opposite electrical charge. The electrochemical gradient is the electrical and chemical gradients combined. So what this means, that ions will flow down their concentration gradient toward an opposite charge, or they will flow down their concentration gradient. If there's more of one ion in one spot, they'll go to an area where there's less ions. So that's what the electrochemical gradient is. So now we get to generating your resting membrane potential. And again, this is talking about the inside of the axon or the neuron, and it's describing the charge that is on the inside of the neuron at resting. So when a neuron hasn't been stimulated and it's just resting, it has a resting membrane potential of negative 70 millivolts. So that's what all your neurons are at when they're resting. They're so negative inside because for the most part, your neurons at rest are just more negative inside of them because of the ions that make them up. So they have a negative 70 charge on the inside. The membrane is said to be polarized because it's at negative 70. And we can actually measure this potential using a voltmeter if we wanted to. Okay, I'm gonna skip this slide and I'm just gonna show this picture. This is showing the voltmeter, showing how the inside of this axon is at negative 70 millivolts. The outside is more positive, but just get in your head that at resting, the neuron has a negative 70 millivolts of resting. So at resting, your nerves are at negative 70. The differences in ionic composition, this ECF and ICF stand for extracellular fluid and intracellular fluid. And I think there's a mistake here in this slide. I'm just reading through this. 1 second. Nope, it's right. Okay. All this means is that outside of the cell, there's a higher concentration of sodium ions. So if you remember anything from tonight, remember that resting potential of the inside of your neurons is negative 70. Outside of your neurons, there's more sodium ions, which is Na positive, and inside your neurons, there's a greater concentration of potassium ions, the K plus. So sodium is always more concentrated outside of the neuron, and potassium is always more concentrated inside the neuron. It's important to remember that because it'll help you remember which way the ions flow when a nerve is being stimulated and an action potential travels through it. This describes resting membrane potential, the animation. This is showing what a typical neuron looks like at resting membrane potential. It shows the general concentrations of your sodium and potassium inside and outside of the cell. You'll notice that both sodium and potassium channels are closed at resting, and outside of the cell, the sodium concentration is higher, and inside the cell, the potassium concentration is higher. And these concentrations just help to establish that negative 70. I know you might be thinking, well, they're both positive. Why is there still a negative 70 charge inside the cell? That's because there are other ions and proteins, mainly large proteins within the cell, that are keeping it at negative 70. So it's not just potassium that's doing that. Okay, what else? Here's some differences in the plasma membrane permeability and how sodium and potassium can leak through and how well they can leak through. The sodium potassium pump is an active transport pump that I think we talked about active transport in your first kind of unit. The sodium potassium pump helps to stabilize and establish this resting membrane potential. And how it does that is that it maintains concentration gradients for sodium and potassium. That means that it pumps sodium out of the cell and it pumps potassium into the cell to try to keep sodium out and potassium in. We want those concentrations to be that way for a resting neuron because a nerve can be more easily stimulated if there's more sodium outside of it and more potassium inside of it. And we'll talk about why. You should know the sodium potassium pump pumps 3 sodium out of the cell, while at the same time, 2 potassium are pumped back in. So if you take your hands and make 1 with 3 and 1 with 2, 3 is Na, sodium, the 2 is potassium, K. The sodium potassium pump just goes like this. It pumps 3 sodium out and 2 potassium in. So you should know that kind of change and how it pumps sodium and potassium into and out of the cell. So here's a look at resting membrane potential. I think this is showing the sodium potassium pump. If It's really hard to see, guys. Sorry, that's really low, unless my eyes are just bad. Step by step. Okay. Changing the resting membrane potential. So now we're going to talk about how you stimulate a nerve, and I won't go too much farther. We'll just kind of try to get a little bit through this. Membrane potential can change, so that charge difference can change when concentrations of ions across the membrane change. So the membrane permeability to ions change. This means that certain channels will open to those potassium and sodium, and they will flow through those channels, and that'll change the charge of the membrane potential. The changes produce two types of signals. They can be graded potentials, and a graded potential is an incoming signal that operates over a short distance. Graded potentials usually occur in your dendrites, in your cell bodies, and they can go in both directions, but they're much shorter lived. The action potential is a long distance signal that is in the axons. Changes in the membrane potential are used as signals to receive, integrate, and send information. So we get into terms describing the membrane potential changes relative to their resting membrane potential. So when we talk about depolarizing your membrane, that's a decrease in membrane potential, which moves it towards 0 and above. So depolarization takes us at resting, which is -70, and it moves us up and creates a more positive membrane potential inside. Inside of the membrane becomes less negative than the resting potential, and the probability of producing an impulse increases. So this is depolarizing. Another way to think about it is depolarizing is taking something away from normal. So depolarizing will be what we want to happen if we're trying to produce an impulse in our nervous system. Hyperpolarization is an increase in membrane potential away from 0. So if your axon becomes hyperpolarized, that means it becomes even more negative than resting. So we're talking about -90 or -100. During hyperpolarization, it's much harder to produce an impulse in an axon because it's that much further away from resting. So here's a look a little bit. So we're measuring the inside. This is going to be talking about a graded potential, but this is resting potential. And if we stimulate the dendrite in this location, we would get an upswing in the graph as that dendrite is becoming depolarized. So its electrical charge is changing, it's becoming more positive. This is a hyperpolarizing stimulus that makes the membrane potential more negative. So again, a graded potential, it's short-lived, it's localized changes in your membrane potential. The purpose of a graded potential is to eventually trigger an action potential to occur in an axon. So graded potentials will happen when a dendrite or a cell body receives a stimulus. So it'll be short-lived. All it will try to do is get that signal or that membrane potential change down to the axon terminal to try to send out an action potential through the axon itself. So here's a look at a stimulus. In the middle, a small patch of some sort of probably dendrite or cell body. And the stimulus will cause an electrical charge difference. Basically what it'll do, it'll cause membrane channels, proteins to open so that some sort of ion rushes into the cell and it becomes positive. And that's what causes an electrical signal to pass through. Depolarization will spread from one area of the membrane to another in graded potentials. So in graded potentials, they'll spread out from the initial stimulus and they'll go in both directions. The current will flow, but it'll dissipate quickly and it will decay. So these graded potentials just don't last very long. So this just is showing you the active site, the initial site of depolarization of a graded potential, and then how the distance won't be that far out. It'll be very short-lived. Action potential. Are you guys still with me or do we need a break? Just gonna keep going. Try to get through action potentials and then we'll stop here. So action potentials and graded potentials will be, if you do go to a workshop, hopefully it's helpful. Thanks, Erin. So an action potential is the principal way that neurons send signals. So this is the way that your neurons communicate. An action potential travels down an axon. It will only occur in muscle cells, which we talked a little bit about last time, and axons of neurons. There's a brief reversal of membrane potential with a change of voltage of about 100 millivolts. An action potential does not decay over distance. An action potential is also referred to as a nerve impulse. So this is just the way that some sort of signal, when a nerve is stimulated, gets passed down. And an action potential, in order for it to pass down and create an electrical charge difference of the membrane, we need to open specific voltage gated channels in order. So here's the action potential. The key players are the voltage gated sodium channels and the voltage gated potassium channels. They can be open or closed if it's a sodium channel, and it can also be inactivated if it's a sodium channel, and that will be important to know. Potassium channels can just be open or closed, but these are the two main channels that we're going to be talking about because they're the two main ions that we're talking about. And this shows a picture already of where sodium will be more concentrated outside of the cell. Remember, I told you to remember that. Potassium is more concentrated inside of the cell. So when one of their channels opens, they will flow down their electrochemical gradient. So sodium will always travel into the cell. Potassium will always travel out of the cell. At resting state, so we're going to go through the 4 main steps of an action potential. At resting state, all the channels are closed because the nerve has not been stimulated yet. Remember, when a nerve is stimulated, that stimulus opens up these channels so the ions can rush in and out, and that's what causes the signal to pass through. But at resting, everything is closed. There might be some leakage channels open to help maintain the resting membrane potential. So let's say it's at -71 or -68 millivolts. There'll be some leakage channels that pop in and out an extra positive charge to keep it at -70. Each sodium channel has an activation gate and an inactivation gate. The activation gate will be closed at rest. The inactivation gate will be open at rest. It'll block again once it's been open to prevent more sodium from entering. And again, that'll be important to know that sodium has an inactivation option. And then potassium channels just have one voltage sensitive gate that'll be closed at rest, and it'll open slowly with depolarization, and we'll talk about why that is. So at resting state, everything's closed. So in our little graph showing an action potential, and you'll see this when you do the workshop, you guys should just do the workshop or at least watch the video and then fill out the handout. You could maybe still turn it in if you don't go to workshop, but pretend I didn't say that. And you might still get credit for it, but just watch that video because that'll help hopefully. So here's at resting state. Everything's closed. The nerve has not been stimulated yet. It's at resting. So in a graph, this line represents resting. And if we would label this line, if we had millivolts on the y-axis, this line would be at -70 millivolts, resting. Depolarization. We've stimulated the nerve, so depolarization is going to occur in that nerve. Sodium channels open during depolarization. When sodium channels open, sodium rushes into the cell down its gradient because everything goes from a high to low concentration. This happens naturally or passively without energy. This sodium activation, the activation gates open, so everything rushes into the cell. Sodium rushes into the cell. This causes sort of a positive feedback response in the neuron. As sodium rushes into the cell, it causes even more depolarization, which opens up more sodium channels. And as a result, the intracellular fluid, or inside the neuron, becomes less negative. And if enough sodium rushes in to bring our resting membrane potential up to -55 or -50, we call this magic point the threshold. And if we can get the inside of the cell to threshold, we'll generate an action potential no matter what. And positive feedback will continue to cause all of the sodium channels to open. So basically, we need a stimulus strong enough in order to send a signal down a neuron. We need a stimulus strong enough to depolarize a neuron up to threshold. This results in a large action potential spike. So reaching threshold in a neuron is like knocking down one domino. Once we reach threshold, we'll reach an action potential spike no matter what. And that means the nerve has fired, or the nerve impulse has generated, and it will pass down the axon. So that's what action potential means. It's a firing of the nerve. We've generated enough depolarization so that this electrical signal or the stimulus will pass through. The membrane polarity, the membrane voltage, will jump to about 30 millivolts. So this is showing the second step. This is depolarization. We've gotten a stimulus strong enough to reach past threshold. And if I was getting into a little more detail on this graph, I would put a dotted line about right here where I would say threshold was. And you could have a failed stimulus that's not strong enough to pass a signal through the axon. But this number 2 refers to depolarization. The sodium channels have opened, sodium rushes into the cell. Potassium channels are still closed at this point. So why does this become positive? Well, if we're at -70 and we get a lot of positively charged ions coming into the cell, all of a sudden the inside of the cell becomes positive. And this is the action potential spike at the top. After we've reached the action potential spike, we go back, we try to get everything back to normal because the nerve wants to go back to resting. And repolarization means repolarizing back to resting. Sodium channels will become inactive and potassium channels will open. The sodium channel inactivation stops the action potential peak from rising any farther. We don't need our action potential to rise indefinitely. We just need the neuron to fire and then pass on its signal to the next neuron. So the sodium channel inactivation gates close, and during repolarization, your potassium channels open. Potassium channels also follow their electrochemical gradient. Potassium is more concentrated inside the cell at resting, so potassium will just exit the cell naturally, passively. When that happens, we're losing a lot of positively charged potassium ions. So that brings the inside of our cell back down to -70. And that's what repolarization means. So here's the third step, repolarization. We have an inactive sodium channel. So we stop that action potential peak from rising indefinitely. Repolarizing means that potassium channels open, potassium exits. We're losing a lot of positive charges inside our membrane. So we come back down to negative again. Because again, this graph always refers to the electrical charge on the inside of the membrane. Hyperpolarization refers to the point in time after an action potential where some potassium channels remain open a little too long or longer than they should, but it's all for a reason. Sodium channels has reset at this point, but some potassium channels remain open, allowing excessive potassium efflux. So this means that too many positively charges are leaving. And what that just will cause the cell to do is get extra negative. And that's what hyperpolarization is. So here's the hyperpolarization dip. And again, that refers to the sodium channels remaining open a little bit too long. They'll leak out excess positively charges, so we get into this hyperpolarized state. Okay, and those are the 4 steps of the action potential. And these kind of next few slides take you through the 4 steps in general. And this is a great slide to look at the 4 steps and what happens at each stage. And when I say what happens, I mean what channel opens and what ion flows through it. This is a great animation of an action potential that you again can download the PowerPoint and watch. The last thing we'll probably just touch on is repolarization will reset the electrical conditions, but it won't reset the ionic concentrations or the ionic conditions. So that's where we need the sodium potassium pump. And we need the sodium potassium pump to reestablish our concentration gradients of sodium outside of the cell. Because remember, all of our sodium rushed into the cell, and all of our potassium left the cell. So we need this active transport sodium-potassium pump to reestablish our electrochemical gradients of sodium outside the cell, potassium inside the cell. There are thousands of them on an axon. And again, they pump 3 sodium out, 2 potassium back in to reestablish those concentration gradients. And I think this is a look at the sodium potassium pump that will reestablish that resting membrane potential. And I think we'll stop there. This kind of goes on with threshold, which we'll start with threshold at next time, because this will kind of review the action potential as we talk about threshold. And next time we'll finish this slide on action potential and finish part C in chapter 11. Stop the recording now.